Glycolysis is the breakdown of glucose. It starts with one 6-carbon sugar. We need 2 adenosine triphosphates, or ATP, and phosphofructokinase. So first, 1 phosphate from each ATP will be added to the sugar molecule. In this step, phosphofructokinase adds a phosphate to carbon 1 and carbon 6, and we now have a 6-carbon diphosphate. It's considered energized because it has the phosphates on the end. Now, the 6-carbon sugar becomes 2 3-carbon sugars, and through the process of about 12 enzymatic reactions, these will become 2 pyruvate molecules. If our reaction occurred in the presence of oxygen, then we'll end up with 2 pyruvic acid molecules, and if not, then there'll be 2 lactic acid molecules. We also will have 2 NADHs and 4 ATPs, so that's a net of 2 ATPs because we used the 2 in the beginning. We just finished glycolysis, which occurred in the cytoplasm, so now we move on to the Krebs cycle, which will be in the mitochondria. This is the 3-carbon pyruvate that we just created. Pyruvate will lose one of its carbons as it binds to oxygen to create CO2, and we're left with a 2-carbon molecule called acetyl-CoA. So once we have our acetyl-CoA molecule, it is able to start the Krebs cycle, and so the 2-carbon acetyl-CoA will combine with a 4-carbon molecule and give us a 6-carbon molecule, and this is called citric acid. So the rate-limiting enzyme in this Krebs cycle is called isocitrate dehydrogenase, and it removes hydrogens. So it helps make sure that NAD+ gets a hydrogen and ends up as NADH, NADH being the reduced version of NAD+. So every time we want to reduce this molecule, so from a 6-carbon molecule to a 5-carbon molecule, we need to lose one of those carbons, and it'll bind to oxygen and be lost as carbon dioxide. From here, this happens again, another NADH is formed and another CO2, plus we get an ATP right here. Then we end up with this 4-carbon molecule. More oxidation reduction reactions occur, and we end up with FADH2 and NADH. And then we end up back where we started with the same 4-carbon molecule from the beginning. So that's one turn of the Krebs cycle, and actually for 1 molecule of glucose, we'll be able to go through the Krebs cycle twice. So the products of the Krebs cycle per 1 glucose molecule will be 2 ATP, 2 FADH2, and 6 NADHs. So the electron transport chain is the last step in all of this. So the NADHs that were just formed in the Krebs cycle are now going to be used, and what happens is they will give up their hydrogen and they will become NAD+ again. Their hydrogen contains a proton and an electron, so the electrons will move through this chain while the protons get pumped to the outside of the mitochondria. Then as these electron carrying proteins go down the chain, you get to this last protein, and a pair of electrons is sent out so that it can be paired with oxygen, and it forms H2O. And this reaction is catalyzed by the rate-limiting enzyme cytochrome oxidase. And then finally we have this enzyme ATP synthase here, which will take in these protons, and then the energy from this will allow an ADP to be combined with an inorganic phosphate, and we have ATP produced.